Hello and welcome to this webinar. Today we will talk about allergen exposure assessment and particularly the essentials of sample collection and immunoassays. My name is Eva Maria King and I am the head of the immunoassay group at Indoor Biotechnologies. Over the course of this presentation, I will address a number of different aspects related to allergen measurements. First, I will talk about applications and relevance of measuring allergens. Following sources of important indoor allergens, I will address sample collection strategies and devices. The majority of this presentation will discuss immunoassays for quantitative allergen detection with a focus on methods, comparisons, and quality control. Allergen measurements form a central part of a wide range of applications, and some of those I have summarized on this slide. Studies of gene-environment interactions in allergic sensitization are one such application, as are occupational health and safety monitoring, exposure monitoring during allergen immunotherapy trials, indoor air quality evaluations of homes, schools, and workplaces, and in this context, the pre- and post-remediation evaluation is a particularly important point. Monitoring of allergen avoidance programs is another such, as is laboratory animal allergy monitoring. Quality control and standardization of allergy vaccines and diagnostic products is particularly important in a setting of allergen manufacturers, as is the efficacy testing of allergen-controlled products and devices. In 2007, the National Heart, Lung, and Blood Institute published guidelines for the diagnosis and management of asthma. In these guidelines, they published that allergen exposure increases asthma symptoms in patients with allergic asthma, and based on that statement, recommended that patients who have asthma should reduce exposure to allergens to which they are sensitized and exposed. And the exposure level is something that cannot be determined quantitatively by visual inspection. They need to know that effective allergen avoidance requires a multifaceted, comprehensive approach, and consider allergen immunotherapy treatment. Allergens are produced by a wide variety of organisms. The most clinically relevant indoor allergens are produced by the dust mite species Dermatophagoides terranissimus and Dermatophagoides farinae, but also cats and dogs, as well as various household pets such as mice, rats, and cockroaches. Molds also produce a variety of allergens and biomarkers that may be associated with health risks. The National Survey of Allergen Exposures in U.S. homes evaluated 831 housing units in 75 locations across the United States. The study was conducted by the National Institute of Environmental Health Sciences in collaboration with the Department of Housing and Urban Development. Based on their results, it was estimated that 23% of U.S. housing units contain high levels of group 1 dust mite allergens. And a high level is defined as more than 10 micrograms of dust mite allergen per gram of dust. Cat and dog allergens were found in all homes, and this was the fact disregarding whether or not there was actually a pet in the home. Levels of mouse allergen that are associated with sensitization were found in 22% of the homes. The study was published by Arps and colleagues in the Journal of Allergy and Clinical Immunology in 2004. Samples for the detection of allergens may be collected in a variety of ways. The method most commonly described in the scientific literature involves the collection of reservoir dust samples. That means settled dust is collected by vacuuming bedding, carpets, and furniture. In contrast, the measurement of airborne allergens is typically produced through the collection of air filter samples. These two methods, however, are not exclusive, and other applications may call for different techniques. Therefore, fabrics, surface wipes, or nasal samplers, to name but a few, may also be appropriate depending on the study design. Disregarding the collection method, all sample types are extracted and then tested for their major allergen content using immunoassays. The Allergen Analysis Service Laboratory at Indoor Biotechnologies receives various collection devices for settled, dust, or airborne samples, some of which are shown here in this slide. While all of them may be used to collect allergens, I would like to point out that spore traps are not suitable for the collection of allergen samples. Indoor Biotechnologies has developed and patented a device specifically designed for the collection of settled dust samples. The DustStream dust collector attaches to most household vacuum cleaners and enables the simple collection of reservoir dust samples with minimal dust handling involved. The DustStream is comprised of a nozzle, an adapter, and has replaceable filter inserts for cost-effective collection of multiple samples. It also has caps that enable optional sample extraction and storage. When reservoir dust samples arrive in the lab, they are processed following well-established procedures. First, samples are sieved to remove larger particles and fibers. Then 100 milligrams of fine dust are weighed out and extracted at a concentration of 50 milligrams per milliliter phosphate-buffered saline containing the detergent Tween 20. Following a 2-hour extraction at room temperature on a rocker, samples are centrifuged. Only the supernatants are used in immunoassay. And this extraction procedure is identical for all allergen immunoassay methods described in this presentation. A detailed extraction protocol can be found on Indoor Biotechnologies' website at the link shown below. Other applications require the determination of airborne allergens. This is particularly the case in occupational health monitoring and studies of laboratory animal allergy. These applications commonly use a short-term sampling protocol in which animal technicians wear IOM samplers that collect airborne particles within the technician's breathing zone. Samples are usually collected on 25 millimeter glass fiber filters at an airflow rate of 2 liters per minute for the duration of a specific task. These tasks tend to be between 10 and 15 minutes per sample. This particular application requires allergen detection technologies with superior sensitivity in order to detect short-term airborne exposures. Maria, which I will describe in more detail over the course of this presentation, enables this type of testing. It allows the detection of mouse and rat allergens at levels below 0.04 nanograms per filter using this sample collection protocol. Over the past 20 years, allergen measurements have been performed using a technique called Enzyme-linked Immunosorbent Assay, or short ELISA. ELISA is based on the capture and detection of allergens using monoclonal or polyclonal antibodies that specifically bind to certain allergens. During the assay, a sandwich is built. First, the microwell of the ELISA plate is coated with an allergen-specific capture antibody. Next, the sample is added. If the allergen is present, it is captured by the first layer of specific antibodies. Following a wash step, a secondary antibody is added. This detection antibody is also specific to the allergen and binds the allergen already captured. A marker on the detection antibody then serves as a linker to a reagent that produces a measurable color change reaction. This color change is then used to quantify the amount of allergen present in the sample. This technique is solid and reliable, and many laboratories have the instrumentation necessary to read ELISA plates. But ELISA has a limitation. It can only be used to measure a single allergen at a time. Only one surface is available to bind capture antibodies, and without distinguishable surfaces, only single analytes can be detected. In recent years, Indoor Biotechnologies has developed new technology, the multiplex array for indoor allergens, Maria. Maria enables the simultaneous detection of multiple allergens. It produces quantitative data, improved standardization and efficiency, and is versatile with regards to analyte selection and customization. It is suitable for the measurement of allergens and molds. In Maria, the limitation of the lack of distinguishable surfaces that I described previously for ELISA is solved through the use of multiple colored beads. Maria is based on Luminex XMAP technology and uses 5.6 micron polystyrene beads with different internal fluorescent dye ratios. These internal dyes create up to 100 distinguishable bead sets. Different bead sets are then covalently coupled with different specific capture antibodies. And the bead sets then form the basis of antibody-allergen antibody sandwiches that are very similar to what I just described for Eliza. The difference is that multiple specific detections take place simultaneously, all in the same reaction well. Once the immunoassay steps are completed, the suspended beads are drawn into the path of a flow cytometer. There they pass by two different lasers. A red laser identifies the internal bead color and thereby classifies which allergen is captured on this particular bead set. A green laser quantifies the signal intensity on the surface of the bead, which indicates how much allergen has been captured. The combination of these two signals provides quantitative information on all allergens included in the array. Currently, Maria can simultaneously detect 11 allergens, and Indoor Biotechnologies continues to work on expanding the panel of available allergens. Eliza and Maria provide comparable results, and this was demonstrated by Earl et al. Differences between the methods lay primarily in their sensitivity. In this table, we compare the lower limits of quantitation for each of the 11 allergens in Maria and Eliza. As you can see, Maria is significantly more sensitive than Eliza, and this difference can be up to 80-fold, as is the case of the Timothy grass pollen allergen Flurry5. Having said that, this high sensitivity is not necessarily relevant for all applications. Eliza's sensitivity tends to be sufficient for most studies involving settled dust. Other applications, however, require the higher sensitivity levels achieved in Maria. In addition to the greatly increased sensitivity, Maria offers other advantages over Eliza in terms of its assay efficiency. Allow me to demonstrate this using an example. Analyzing 200 samples for 8 allergens by Eliza requires running 96 Eliza plates. This may take a trained technician between 10 and 16 workdays depending on individual capabilities. Using Maria, all allergens are measured simultaneously, so the number of allergen in the mix does not affect assay time. In our example, all samples are measured on a total of 10 plates, which are typically analyzed within 3 to 5 days. Using Eliza, each additional allergen adds 2 technician days to the study. The timing remains the same for Maria, as all allergens are always measured simultaneously. Indoor Biotechnologies offers a wide range of immunoassays for allergen detection. Eliza kits and analysis services are available to detect allergens of dust mites, cat, dog, mouse, rat, cow, horse, German cockroach, pollens, molds, and foods. Maria kits and services are currently offered for allergens produced by dust mites, cat, dog, rodents, German cockroach, pollens, and Alternaria molds. At this time, more allergen assays are available for Eliza than Maria, but Indoor Biotechnologies' assay development team continues to add more analytes to the Maria panel. Since Maria was first launched in 2008, Maria reagents have been available as fixed 5-plex or 8-plex kits to include the most clinically relevant indoor allergens. Starting in fall of 2012, Indoor Biotechnologies now offers all Maria kits in a new form. Using the new Maria Pick Your Plex, customers can now select any combination and number of available allergens to create their optimal kit. Maximum flexibility through full customization. Both Eliza and Maria are valuable assay techniques. Each has its sweet spot in terms of applications. I would like to give a brief overview over the particular advantages. Eliza measures one allergen at a time. Its sensitivity is sufficient for most studies involving settled dust samples, and results correlate with Maria. Indoor Biotechnologies' Eliza reagents have been used in over 300 publications. Click on the Google Scholar link on this slide for a complete list of Indoor Biotechnologies citations. The reader technology required to analyze Eliza plates is present in many laboratories already. And even if that's not the case, the upfront investment is relatively low. Eliza is therefore a good option for labs with limited resources. It is ideally used for studies involving only 1 to 3 allergens. Maria measures multiple allergens simultaneously. It is a high-throughput technology, state-of-the-art, and offers superior